Hey everybody, we'll go ahead and get started. So today, just as a reminder of where we've been, the last couple of weeks we've been focusing on kind of getting some background on the science behind e-flows. We started with an e-flow overview. We talked about geomorphology, watershed morphology with Dr. Pasternak. Peter came last week and talked a little bit about fish and flows. And this week it's my pleasure to introduce Dr. Eric Stein from the Southern California Coastal Water Research Project, which is a huge mouthful. So most people typically say SCWRP. And he'll tell us a little bit about that agency. And he's going to be talking about the relationship between flows and ecology. He does a lot of work with stream benthos and macroinvertebrates, particularly down in Southern California. And he's going to start this transition of how environmental flows science gets put into policy and management. And so that's what we'll hear about today from Eric. And then we'll move into panel discussions in the next couple weeks, which will focus on particular topics related to policy. So really quick before Eric starts, afterwards we're going to do pizza with Eric after, if anybody would like to stay, free pizza. So I would like a show of hands of how many people are interested in pizza after.
Raise them all high.
Hi.
Pizza. 1, 2, 3, 4, 5.
Okay, so small. Okay, thanks, Megan. Okay, so thanks. So we'll do that right after. We'll head over to Viemeyer. Everybody's welcome. Come, grab a piece of pizza, talk a little bit, and then head out. That's great. Okay, so it's my pleasure to introduce Eric. Thank you very much for coming, Eric. And then you will have questions after, it sounds like, for a little bit after. Okay, thank you.
Thanks, Sarah. I'm happy to be here. And so, if during the course of when I'm speaking, if you have like a burning question that I really don't understand what the hell you just said, just raise your hand because I don't want you to sit there wondering about it. So, we can keep it small group. We keep it informal. So thanks. I'm really happy to be up here and share some of the work that we've done in Southern California and build on some of the work that we've done with the Center for Watershed Sciences here. So I'm going to start by telling you a little bit about my organization because you might not be familiar with SCWRP, and so I'll give you just a real 1 or 2 slides on that, and then we'll just jump right into the fun stuff. So SCWRP is what's known as a joint powers agency, and so if you're not familiar with joint powers agencies, it's basically a public entity that's formed when a bunch of other public entities get together and say we have something we want to do in common. We have a common goal or objective, and we don't all want to do it on our own, so we're going to form some sort of regional agency to do it for us, and that's what we are. And so our joint powers was formed in 1969 in response to the State Port of Cologne Act, which is the state's Water Quality Act, which predates, it was actually the model for the Federal Clean Water Act. And so it was initiated largely by a bunch of wastewater dischargers who said, we're about to be regulated on this new set of regulations that are going to prevent us from discharging untreated sludge into the ocean. And we have no idea even what the baseline condition of the resources are or what the technical questions are. So they formed SCWRP to answer some fundamental questions. We were formed initially as a 3-year project. That's why the last P in our name is project. And like any good scientific report, the last 2/3 are all the things that we don't know. And so they said, we'll keep you guys around for a little while longer, right? And so that's been almost 50 years ago now. But our composition has changed over the years. Our member agencies were a little bit unique in the sense that our joint powers agency now has sort of half regulatory agencies. So these are agencies that write permits and policies, and the other half are discharger agencies, wastewater and stormwater agencies. So these are the people who have to comply with the policies. So we sort of sit between the regulators and the dischargers and really conduct a lot of the fundamental science that they'd use to inform a lot of their policy decisions. We also have agencies at the federal, state, and local level. So we're sort of working across all 3 levels of government. So it's a fairly unique model, and it's a really interesting sort of place to work. So this is I put it up here, these are our major technical departments. I head up the biology group, but we cover, so I'm going to be focusing a lot on biology and hydrology, but we have groups that cover other topics as well. And then so fundamentally, what we do on a day-to-day basis is we do a lot of fundamental research. We do almost all of our research is done in cooperation with universities like Davis, as well as other agencies. We do a lot of work on transitioning science into implementation. That's probably our strongest niche, is working directly with the agencies to facilitate the transfer of science into implementation and policy. And so we don't have any regulatory authority, we don't set policy, but we try to inform policy with our science. And then one of the other things we do, which I'll talk about today, is we coordinate a lot of the regional monitoring efforts in Southern California on both the freshwater and marine side. And so that's part of our role, is to facilitate and coordinate those programs for the region in terms of getting the surveys done, implementing them, and managing all the data. So that's a little bit about my organization and what we do. All right, so let's get into the fun stuff now. So I'm going to be talking about environmental flows and specifically how environmental flow science translates to informing policy and management decisions. And so going back, I know you've heard some of this before, particularly probably from Sarah and Greg in their talks, but just to kind of come back and recap, we know that fundamentally in all over the world, but particularly in California, there's been a lot of fundamental changes to the landscape since people have come in, and so this sort of conceptualization of the greater LA basin shows that we've gone from watersheds, which were sort of highly pervious with lots of streams and wetlands and open space, to things that look like this, where you sort of have fundamentally converted the landscape. And so we know that when you do that, you get this concept of hydromodification, where we change the runoff characteristics, and so we fundamentally change the whole the way water flows across the landscape. So as we go from a more natural to a more built environment, two fundamental things happen. I mean, I know you've heard this before. We sort of change the water part of the equation. So we go from sort of these lower, more drawn out types of peak flows spread over a period of time to these more extreme events where you have higher peaks and the peaks are compressed and you have these sort of flashier types of runoff events. And that's usually coupled with a change in the sediment regime. So usually what happens is you have sort of more water coming off with more energy, and you usually have less sediment because you've paved over a lot of your sediment sources. And so that combination of more water and less sediment leads to fundamental changes in the channel, often either deepening or widening of the channel. That's sort of what we refer to as hydromodification. So it's that combination of changes in both the water and sediment regime. And so, this is some data. This is from one of our study sites in Southern California. So just to quantify some of this, the effect of this hydromodification is that things like this is the instantaneous peak flow in this watershed as a function of recurrence interval. And so in a pre-development stage, we can see that a 10-year event had about 2,000 CFS in terms of instantaneous peak flow, whereas in the post-development, that was up closer to about 7,000 CFS. Or another way to look at this is instantaneous peak flow of 4,000 CFS would normally occur probably every 35 to 40 years, now occurs every 3 to 5 years or something like that, right? So those are the fundamental effects. And the other thing I want you to notice from this is not a very developed watershed, right? Only less than 7.5% impervious cover. And so in Southern California in particular, the nature of our landscapes is we have a lot of really young geology, especially in the coastal plains of Southern California. So these are old marine sandstones, and so they tend to be fairly poorly consolidated. The soils are really young, so you have this really unconsolidated type of substrate. You also have very steep watersheds. I often use the analogy in our area in Southern California, you go from about 3,000 meters to the ocean, to sea level, in about 150 kilometers, right? So that's a really steep, in terms of something like that. That same elevation gain is probably more than what you cover in the entire length of the Mississippi River, right? Over, many, many, many more stream miles. So we have these really steep watersheds, high precipitation, high intensity rainfalls, falling on steep watersheds with very poorly consolidated substrates. And so when you put all of those together, you sort of get a recipe for some fairly dramatic effects. And so this is an example of one of our study sites in the Lancaster-Palmdale area. So if you're familiar with Southern California, this is sort of north of Los Angeles, and this was supposed to be the next suburb that nobody moved to, right? And so you have a lot of this kind of low rural residential. So this area, this is about 2 to 3% impervious cover. And if you sort of read the literature, what it'll tell you is, oh, that's pretty low. You shouldn't expect to see a lot of geomorphic response at just a couple of percent impervious cover. But this is what we see, and this is the channel that I was just showing you. I was asking Sarah if she knew Derek Booth. That's Derek Booth sitting there. He was helping me do the field work. But this is a fairly dramatic response. And if I show you the cross sections, this is what, in 1990, this is what the pre-development cross-section looked like, this red dotted line. And some of the contemporary cross-sections, you can see, with only a 2 to 3% impervious cover, you see really dramatic responses. And so it's this combination of all the factors that I just talked about produces these really dramatic responses. So that is actually a real problem from a management perspective, right? Because it means that these landscapes are highly, highly sensitive. And so if you sort of sneeze on them, they could fall apart, right? And so it raises some challenges that I'll talk about later in terms of how you allow things to happen from a development perspective and try to protect against these types of effects. All right, so if we think about, now that's kind of just a little backdrop of sort of the world I work in, and I'll come back to that in a second, but I want to scale back up and say, so what does this mean in terms of kind of the overall management questions that we're trying to answer? So I gave you a few examples from Southern California, but we know statewide, this graphic comes off of a recently published paper, and it's one of the first attempts where we tried to look statewide and sort of ask the question, how much hydrologic alteration do we have within the state of California as a whole, right? And so that's what this paper did, and it looked at gauge data throughout the entire state. And what you're seeing here is that we see this pattern of depletion of high flows. So the general patterns we see is we tend to see these depletions of high flows, and so this is due to things like damming and water control structures, and we tend to see this augmentation of low flows, and a lot of that's due to urban runoff, agricultural return flows, and things like that. And so this combination means that we have more than half of the gauge locations by the criteria used in this paper are considered altered. So we have a fairly extensive amount of hydrologic alteration. It's a fairly pervasive issue in the state of California. So we know that this is a big problem. One of the questions is that's fine, right? But from a policy or a management perspective, you sort of have to ask the question, well, how much is too much, right? How much flow depletion is too much flow depletion? Or how much augmentation of base flows is too much? So that's really, when we talk about environmental flows and ecohydrology, that's really fundamentally the question we're trying to answer. How much is too much, and how do you make those types of decisions? And so we need an approach, or we need a way to define what do we mean by flow impairment. When have we passed some sort of critical threshold where it's too much alteration, and how do we make that decision and use that to trigger certain management actions that we may want to impose to either try to reduce or remediate those types of effects. Now, we're fortunate in California because the state has actually invested quite heavily in biological monitoring over the course of the last 15 or 20 years. And so when I go in other parts of the country and I'm talking to people and I show a map like this, they're like, oh my God, this is like a gold mine, right? Because this type of data density doesn't always occur in many other places. And so what this map is showing, this is a program administered by the State Water Resources Control Board called the Perennial Stream Assessment. This has been going on for about 15 or 18 years now. And this is a probabilistic survey. So these are randomly selected sites where every year field crews go out and they measure a whole bunch of conditions at these sites. Primarily, they look at benthic invertebrates, stream algae, and a bunch of physical properties, which I'll talk about on the next slide. But the point of this graphic is that we can actually now mine this data set of biology, right? So this is a data set of biological conditions at streams across the state. We can mine this data set to begin answering that question of how much is too much. Right, so I asked you, well, how do you know to set that impairment threshold? So what we want to do is take advantage of this really nice data set to begin exploring how to answer that question of how much flow alteration is too much flow alteration. And so I'm going to give you an example for where I work down here in Southern California. So this is my office is sort of right there, and one of those red dots probably. And so this is our, in Southern California, you'll notice we have a fairly high data density because we have a local intensification of our regional monitoring program. So we take the state program and we intensify it. So because it's a data-rich area and we have ongoing regional data collection, it's a nice laboratory to begin exploring some of these relationships between flow and ecology, because we have some really nice data sets to work with and test out approaches that potentially could be scaled up to address these issues at a larger spatial scale across the state. So the balance of most of what I'm going to talk about from here forward is really focused on the work here in Southern California, but the goal over the long term is to try to scale this up and apply it more broadly across the state. I'll just point out one other thing. Out here, we don't have any points, right, because these streams are dry. But what I will tell you is that there's a whole nother set of projects going on right now to develop a new set of biological indicators that are more appropriate for these really dry streams. So the hope is down the road, we'll have dots in this part of the map, in these drier parts of the state where we need a different set of indicators. All right, so what are the indicators that we use? This is our stream assessment toolkit. So these are tools that we use throughout the state of California. We have tools that measure sort of the in-stream biology, so the sort of the health of the stream. And there's two of them. There's a California Stream Condition Index, which is based on benthic invertebrates. I'll talk more about this on the next slide. And then we have an analog that looks at in-stream algae, primarily benthic algae. And so these are two really good indicators. I'm not actually going to talk about the algae today in the interest of time, but we use these in concert because the benthic invertebrates as a primary consumer, the algae as a primary producer, they tell us some different and complementary things about the health of the stream. So we use them in concert to give us a more robust picture of stream health. I'm going to focus on the bugs today, though. We also take, at all of our sites, we do some physical habitat measurements in terms of the condition of the physical habitat and hydromodification, and we do a general stream condition survey using a method called CRAM, or the California Rapid Assessment Method, which is sort of a riparian condition assessment tool. I'm not going to talk about that today either, but I just wanted to give you sort of a sense of sort of the suite of data that gets collected at each one of those points on the map. So we have a fairly nice data set to work with. So we like bioassessment, and bioassessment can be done for benthic invertebrates, it can be done for fish, it can be done for algae. I'm going to focus on the bugs today, but the reason we use bioassessment is because really, at the end of the day, when we're thinking about managing streams and managing for stream health, what do we really care about? Is we really care about the biology, right? And so by using bioassessment, it gives us a direct measure of what we're actually trying to protect. The other nice thing about biology is most streams have some level of biological communities, especially the bugs and the algae. Not all streams have really healthy fish populations. It's a little more, especially in Southern California, our fish populations tend to be fairly depauperate, but bugs and algae are fairly ubiquitous across our streams. And the other nice thing we like about them is that they sort of incorporate, or they integrate stressors, right? So the difference between taking, say, a chemical measure, which represents sort of the amount of nitrogen or copper in a stream at a point in time, the organisms tend to integrate all of that exposure over the time that they're living in the stream and give you more of a holistic assessment of the stream health. So for all those reasons, we like to use biology, and in most cases, many of the state programs use bugs and algae as kind of the key biological indicators. So the challenge is, we go out to the field and we run our transects and we collect 1,000 bugs and we subsample them. And so now we have all these bugs and we identify them taxonomically. And so at any given site, you get some combination of bugs, and you get some things like your stoneflies, your mayflies, and your caddisflies, which tend to be indicative of more healthy streams. You have things like your midges. I often refer to midges as like the pigeon of the bug world, right? It's like, when nothing else can survive there, the midges are doing great, right? And so those are sort of more indicative of more stress systems. And then you have some of the ones that are more in between, certain types of beetles and dragonflies, which are a little more variable. So by looking at the composition of who's living in the stream, we can actually learn a lot about the overall health of the stream and what types of stressors might be impacting the stream. But for a manager, I can't really go to a manager and say, well, I've got 100 stoneflies of this species, and I've got 300 midges of this species, and the manager's like, well, what do I do with that, right? And so what we do is we create these scores, or these indices, where we take the composition of the community, we take who's living there and how many of each organism is living there, and we convert that to some sort of scale. It's sort of like a GPA, like you take your grades and you put them into a GPA, and it gives you one number that represents kind of the overall sort of assessment of how you're doing. And so that's really what we do with all of this as well. And so in California, we use what's called the California Stream Condition Index. I'm not going to go into the details of how it's calculated, except to tell you that it's a relative index, which means what we're doing is we're comparing the conditions at a site, what we observe at the site, relative to what we would expect to see at that site if that site were in reference condition, if it were pristine. So we use models to predict what would that site look like under totally pristine conditions. We go out, we measure what we actually see, we compare what we actually see to what we would expect, and the ratio of that observed to expected condition gives us a score. So it ranges from 0 to 1. So 1 means it's in reference condition, it's exactly what we would expect to see under reference. 0 is at the other end of the spectrum, and we have a sort of a putative threshold at 0.79. Right, and that is basically the 10th percentile of the reference distribution. So if we look at all of our best sites in the state, the 10th percent of the reference distribution is at 0.79. So use as a threshold to define impacted versus healthy. Okay, there's nothing, sort of magic about that. It's basically just what the data tells us. I'll tell you that this number at the current time has no policy bearing, right? There's no policy in the state that refers to this number, but in terms of sort of practice, that's what people use as sort of a threshold. And so that's the important thing. It'll come up later when I talk about some of these flow ecology relationships. I'm going to refer back to this threshold. So remember that number, 0.79. That's the magic number, right? All right, so we'll come back to that. And so that's the index we use, the California Stream Index or the CSCI. So if I refer to CSCI, remember that. All right, so now with that background, the questions that we're trying to answer with that with that technical background is we want to go into the region, we want to know how healthy are our streams, we want to know what are the stressors that are most impacting those streams, and in particular, we want to know how important is flow modification relative to other stressors. So if a stream is not in good condition, is it due to flow? Is it because the flow's altered, or is it because there's a bunch of copper in the water, or maybe a bunch of pesticides in the water, or some other stressor? So we want to be able to tease that apart. And then we want to look at the management implications and say, okay, if the stream's not healthy, what can we do to try to, from a management perspective, to try to improve it? Or if it's at risk, how do we keep it from tipping over and getting below that 0.79 threshold? And so that's what we're trying to do with this. And we're going to try to relate changes in flow to that magic 0.79 threshold and use that to inform our decision-making process. So in Southern California, the types of impacts that we're concerned about, I've given you sort of a little, sort of why we do this, right? Besides the fact that it's fun and bugs are cute and you get to get out and get, your feet wet and dirty and all that other good stuff, reasons you become a biologist. But what we're trying to do is, probably this is where I like to start because, even in Southern California, we actually have some fairly pristine areas in the upper parts of our watershed. And so we want to know where those are, and we want to do whatever we can to keep them pristine, right? And the state actually has policies on the books called anti-degradation policies that sort of create incentives or disincentives to keep people from degrading these poor systems. So we want to use it to guide our management decisions and keep our pristine places pristine. But in Southern California, most of what we have, we don't have a lot of sort of big dams like you have in some of the central part of the state. The ones we do have are more for flood control purposes and not hydropower. But we do a lot of stormwater capture. So a lot of our streams have these rubber dams in them where we impound the stormwater and we capture it and we divert it for infiltration purposes. We're also doing a lot of reuse of treated wastewater. So many of our streams are effluent dominated, meaning you have a wastewater treatment plant like this one right here, which discharges treated effluent into the stream, and that makes up the predominant base flow in the system. And so increasingly, we're seeing this type of treated effluent being diverted for recycled water use, as well as stormwater capture being diverted for reuse, right? And so those management actions fundamentally change the hydrology of the streams. And so one of the things we're interested in is, what is the ecological effect of those types of management actions? So even though this stream historically may have looked really different, over the last 75 or 100 years, the ecology of that stream now depends on these arguably artificial flows, right? And so the question is, what do you do about that, right? And so we still want to understand what that is, and we still want to understand, is it, what do we want to protect? And then, of course, now that it's February and it's like summer up here, I don't need to tell you that drought and climate change are big issues, and we expect them to be increasingly big issues. In fact, we already know, because we have 15 or 20 years of data, that the distribution of our species has started to shift because of some of these long-term climate. We can see it in the data, and so we know that the sort of whole landscape is shifting around in response to drought and these extended droughts and climate change. So we have to understand what those implications are, and so when we manage for flows, it has to be done against this backdrop of expected changes with more extended drought and generally warmer, drier conditions. All right. So I just want to reiterate the fact that beyond all of the cool science and all of that, there's lots of policy drivers. So the state has many, many programs in place that even if you don't care anything about those cute little bugs, There are many programs in the state of California that will compel you to care about them if you want to do anything on the landscape, like manage wastewater or build homes or infrastructure or grow crops, right? And so there's lots of regulatory drivers in the state that make you want to care about this, and they all use, or many of them use, the types of tools like the CSCI that I just talked about earlier. All right, so what do we do with all this? So this is a snapshot of our Southern California stream assessment. This represents the first 5 years of data that we collected between 2008 and 2013. And so you can see we get a nice distribution. We can look at, of the 4 indicators we look at how many tell us, do none of them tell us that the stream is in good condition, or do all 4 of them tell us that they're in good condition? So we can get a sense, they don't always agree, obviously. So we can start to get a sense of the condition of the stream from a biological perspective. So we start with that. We have an understanding of kind of the biological condition of our area. Then we want to start where we know that, we have a lot of dots. We have a lot of things that aren't blue here. So in all the areas that are red and orange and things like that, we want to kind of know, is flow the driving factor that's causing those conditions to be impacted, or is it something else? What are the key stressors? So to answer some of that, we do some basic flow measurements, but we rely heavily on these physical habitat measurements. So we go out and we look at substrate, we look at algal cover, we look at in-stream habitat, we look at riparian vegetation. So we do this physical habitat assessment in the field. We do a lot of sort of cross sections and measurements, and that's all done. And we generate a set of metrics, just like we do with the bugs. We generate a set of metrics that speak to sort of the physical condition of the stream. And then we can take those metrics, just like we do with the bugs, and we can convert them to an index. And so just like we have an index for the bugs, we have an index for the physical habitat that we call the IPI, the Index of Physical Integrity. And so this is a map of that same study area, and it shows this index of physical integrity. If it's, how does it break, how does it divide in terms of the percentile distribution? Now, we don't have as much data here, and so we don't have sort of a threshold like that 0.79 threshold. So we're just showing, are they red? Are they in the lowest one percentile of scores, or we top out at the highest 30th percentile? So even the fact that, that number is fairly low tells you that we have a lot of physical alteration, because our best category is even so greater than the 30th percentile. But the interesting thing here is we can start to look at this index of physical integrity, and we can see that the score, again, it tops out at 1 in theory. It can go a little bit above because of some statistical anomalies. But the take-home message of this graphic is that we actually see a pretty good relationship between the CSCI and the index of physical integrity. So we know that at least there's an associative relationship between sort of physical alteration and changes in the benthic community. So that's a good start, gives us some good evidence. The other thing that we do is we do a lot of directed evaluation of that channel incision that I showed you in those opening slides, that hydromodification, where we see that channel down cutting or that channel widening. So we have a tool, we have an assessment tool that looks at sort of the susceptibility or the likelihood of impact of streams in terms of both in 2 dimensions. So this tool looks at sort of the lateral response. We have sort of these indicators and these checklists we look at that sort of rate the stream in terms of hydromodification impact in the lateral dimension. So it's a channel widening. And then we have tools like this. This is a tool that looks at bank angle versus bank height. And we can, by looking at the 2 of those in concert, we've been able to empirically derive this curve. And we can tell if you're on this side of the curve, you're sort of at a high risk of incision, and if you're on this side of the curve, you're at a low risk of incision. So that's an empirically derived relationship from regional data. So we can use that to start to rate these streams in terms of widening or deepening, basically, is what we're asking. So we want to know, is the stream tending to widen or deepen? So when we put all that together, we can produce data that looks like this. Again, the same map. And here what you can see is if we look at the distributions, we can see that we tend to see much more lateral. We tend to see much more widening than we do deepening. And we can see that agriculture is sort of a pretty big culprit. Now, there's a little bit of an anomaly, I'll tell you, is that this urban here, you say, well, urban really isn't that bad, right? The red bar isn't that bad. But you have to remember that a lot of our urban channels are concrete. And so by definition, they're not going to widen or deepen because we fixed them in concrete, right? So there's a little bit of an anomaly in here because there's some of our, even if they're not hardened, they might be armored with riprap or things like that. So there's a little bit of an anomaly in there. But the take-home message is, again, we see fairly pervasive impacts, and we can sort of quantify whether this is a problem in the vertical or the horizontal dimension. So when we put all of this together, we can generate this relative risk plot. And so this relative risk plot basically is just a way to rank the stressors. So we look at all of the data that we collect, and we sort of ask the question, which stressors are most associated with poor biology? And so it's sort of like the same. The relative risk plot is like when they tell you you have a 40% increased likelihood of getting cancer if you smoke, right? That's a relative risk because you're at more risk than a non-smoker. So the way this works is you can say, if you have a lot of phosphorus or a lot of channel alteration or a lot of winnowing of your sands and fines, you have a much higher probability of having impaired biology than if you have sort of high selenium or copper. And so what this tells us is that hydrology and geomorphology are the largest drivers. So if you, as a manager, I can say, you know what, copper is not your big problem. And ironically, you actually have a lot of regulatory programs on the books that limit copper, but that's actually not your big problem. So that means one of two things. It either means, A, your regulations are working because you're regulating it and it's not a problem, or B, you're regulating the wrong things because there are no regulations to deal with things like channel alteration and sands and fines, and those are your big problems, right? So this is really important backdrop before I get into the flow ecology, because what we've been able to sort of prove is that if you're going to invest in management, invest in hydrology and geomorphology, right? Because those are really your big drivers in terms of the biology of the stream, more so than chemistry or kind of your traditional pollutants. So this is actually really important for managers to understand because it's actually a lot more expensive to deal with hydrology and geomorphology, and so, but we actually, I think, have a pretty compelling case that that's where you want to be devoting your energy. All right, so what do we want to do with all that? So at the end of the day, what we really want to do is we want to relate this change in flow to some sort of change in biology. So we want to say how much change in flow is too much change in biology that it gets us outside of this zone. If this is sort of the zone we want to be in, how do we sort of create this curve and use that to set some sort of target that we want to aim for? And we're going to base it on bugs, and we're focused on sort of these urban water management types of issues. All right, so there's lots of ways to do it, and so there's lots of ways to generate those relationships. This is a really nice publication that came out a few years ago that summarized a lot of methods. I actually really like this publication because it's one of the few times that two federal agencies actually got together and produced a joint document and agreed on something. So that was actually pretty remarkable, I thought. So there's mechanistic approaches. This is some of Sarah's work that looks at sort of functional flows and mechanistic. There's also statistical approaches, like some of the presumptive standard work that Brian Richter's done. And so there's different ways you can do it. In Southern California, we use an approach called the ecological limits of hydrologic alteration, or ELOHA. And the reason we used ELOHA is because we thought it was kind of the best of both worlds. For our purposes, we're focused on bugs, we're focused on benthic community, and we want to do something regionally that's based on a community response as opposed to sort of a single species response. And for those reasons, ELOHA was a good fit for our particular application. And so, again, because it's community-based and it's regional versus sort of site-specific and species-specific. And so the basic way it works is you have to be able to look at the amount of hydrologic alteration you have within the system, and you use that to calculate a series of flow metrics based on your degree of hydrologic alteration. You relate that to the degree of biologic change, so you're relating hydrologic change to biologic change, and then you create some relationships that lead to that sort of hypothetical S-curve that I showed you a couple slides ago. And then we want to use that to develop some sort of regional index or target that answers the question, how much hydrologic change and what type of hydrologic change results in an unacceptable biological response, which we define as a CSCI less than 0.79, right? So what type of hydrologic change and how much gets you below that magic number, and that's sort of what we want to try to do with this. And then we want to apply that to inform management decisions. All right, simple, right? All right, so how do we do that? All right, so this is the general process I've sort of talked about already. We started with a statewide classification that we worked on with the folks here at Davis. We go through we compile the regional bioassessment data. We estimate hydrologic alteration using models. we do some logistic regression to try to look at thresholds, and then we pick a set of metrics that are the most important, and then we evaluate our endpoints. So I'm going to walk you through each of those, sort of just one or two slides on each one. All right, so we start with our regional bioassessment data. I've already showed you a different version of this slide. So we say, okay, great. We know where we have good bugs and where we have bad bugs. So now the next problem is, so remember I told you that CSCI is observed to expected, right? It's a ratio of what you see to what you expect. So by definition, it already measures biologic change, right? Because it's deviation from reference, right? It's deviation from expected reference. So I've got the biologic change nailed. So now all I need is the hydrologic change, right? So all I need to do is I need to get a measure of hydrologic change at every one of those dots, right? Simple, right? No, right? Because none of them are gauged, right? These are all little streams. There's no stream gauges on these. So we have to basically go back to models. Right, so what I need to do is I need to go with each of those dots, I need to model the amount of hydrologic change that's occurred at each one of those points from some sort of reference condition to what was there at the time that we took the measurements of the bugs. Well, there's 830 of those dots on the map, right? And so I couldn't actually pay somebody enough to do 830 models, right? So what do? So we developed this ensemble model approach. So we said is we can't do 830 models. It's really not practical. So I developed an ensemble modeling approach. And so what that really means is I look at all of the catchments that are associated with those dots. So I look at and what are the range of types of catchments in terms of the size and the elevation. And I'm going to group those catchments into sort of similar categories. And then I'm going to develop a representative model for each of those groups, right? And so that I create essentially an ensemble of models that represent the region as a whole, and I can use that ensemble of models now to basically model change at each of those 800 or so sites. And so that's basically what this flow chart basically says. And so we went through this clustering analysis. We wound up with 26 models. So we took out of those 800 so sites, we boiled it down to about 26 models. We used a relatively simple modeling approach, an HEC modeling approach, very simple, straightforward. It's got some limitations because it doesn't have all the bells and whistles that some of the more advanced models do, but it's actually fairly parsimonious, and at this type of scale, it sort of answers our basic questions. And so you can see the ranges of catchment sizes and the ranges of impervious cover. So we've got a broad range of impervious cover. We've got different catchment sizes, elevation ranges. And so now we've got these 26 models, and we're able to cluster all of those 800 sites and assign them to one of these 26 models. Right. And so essentially what we do is for every one of these novel bioassessment sites, I go to my library of models, those 26 models, and I say which one of those models is the best fit for that site. And we make that decision just based on similarity of catchment properties, right? So we look at the catchment size, land cover, slope, elevation. And so we have an algorithm, we have a clustering algorithm that allows us to assign each of those novel sites to one of those 26 models and pick the most appropriate one off the shelf. So now I have a more manageable set of models to work with, and I can go ahead and simulate a time series of hydrology under contemporary current rainfall conditions. And I go into the model and I turn the catchment natural. I take all the land use out, I change the routing parameters, change the time of concentration, change the storage properties. I make the catchment look natural in the model. I run it again. And so now I have natural hydrology and I have current hydrology for each of those sites. And the difference is that delta H, or that hydrologic change. So now I have a measure of hydrologic change and a measure of biologic response for each of those 800 sites. All right, so that was sort of the big hurdle to overcome. So I still need metrics, right? So I take the hydrology comes out as essentially hourly, flow, right? And so I take that hourly flow data over the period of time. We actually modeled a 6-year period that represented 2 wet years, 2 dry years, and 2 average years. So I have a 6-year period under reference conditions, a 6-year period with the same rainfall properties under contemporary conditions. And so that's what I use as my delta H. So I calculate a whole bunch of metrics some that talk about magnitude, variability, duration, timing. So I calculate a whole suite of different metrics. and I look at those metrics under average years, wet years, dry years, and all years together, right? So it turns out to be that I have something like 126 different metric combinations that I'm looking at, right? So now I've got all of these metrics, 126 metric. I've got change in hydrology based on all these metrics, and I want to relate them to those biological scores. And so this is the way we did this. This is a logistic regression plot. And so thankfully, things like R, I can actually do this process hundreds and hundreds and hundreds of times in a fairly efficient manner. And so what this logistic regression plot shows is it basically is looking at, this is one flow metric on the x-axis. This is high duration of flow, and it's in days, high duration of flow, so days, high duration flow days. And this is in the decreasing direction. So here's 0. So this is a decrease in the high duration of flow. There'd be another plot on the increasing. The reason I sort of break them is the shape of the plot is really different on the increasing side than the decreasing side. So now it gets even more complicated because you have to look at increasing and decreasing separately. But for this example, I'm looking at a decrease in high duration of flows. And on the y-axis, I'm looking at the probability that my CSCI score drops below that 0.79 threshold. So how much hydrologic alteration for this one metric, high duration of flow, how much of this results in a certain probability that I drop below my 0.79 magic threshold and say that the bugs are in bad condition? Now I picked 50% because it's in the middle, really, right? But there's nothing, again, magic about 50%. So the nice thing, the reason I like these logistic regression plots is because as a scientist, I don't get paid to make a policy decision. I just get paid to provide the tools to help somebody else make a policy decision, right? So I can tell them as a policymaker, you may decide that 75% is what you want, right? You may say, I want a 75% probability, or I want a 25% probability. That's really your choice. You can use this plot, and you can pick whatever probability you want. If you want to be sort of risk averse, or you want to be, want to be more liberal in your decision, you can choose where you want. But for a 50% probability, I can now tell you that this drop, if I decrease the high duration of flow days by about 7 days, so on average, if my high duration of flows drops by about 7 days, then I have a 50% chance of the biology becoming impacted below my magic threshold, right? And so that's really the way these plots work. And so if you wanted to pick 25%, then you come out here and you could say, well, then I can decrease it by more because I'm allowing more impact to occur, right? So we do this for lots and lots of these, right? So we look at all the hydrologic metrics, we look at the CSCI, we look at different permutations of CSCI, so I have hundreds and hundreds of these things. And then I go through a metric screening process, and I sort of start to cull I look at all these logistic regression plots, and I say, which ones affect the in-stream biology the most? I get rid of redundancy, right, because I don't want to have a lot of redundant metrics in there, so I get rid of redundancy. I want to look at things that actually differentiate reference from non-reference, so they have discriminatory ability. And I want things that are amenable to management, right? Because a flow target doesn't help you if you can't do anything about it, right? And so those are sort of the filters. So we run all those flow metrics through those incremental filters, and we wind up with this final list of 6 metrics that we recommend based on all of that analysis. We come up with these 6 metrics as the 6 metrics that we would recommend to managers as the ones that you should be sort of aiming for. If you want to create a flow regime that is protective of the biology as measured by those benthic invertebrates, these are the 6 ones. And so for each one, I can tell you is it most important during average rainfall, wet or dry conditions, and in some cases the threshold's on the decreasing side, and in some cases the threshold's on the increasing side, right? So it kind of depends on the metric whether too much is a problem or too little is a problem. Right. So that's basically the punchline, right? At the end of the day, that's what we come up with is this set of metrics, right? And so then the last couple of slides, I'm just going to show you a few applications. I can put all of those together. I can aggregate them into an overall score, and I can look at degree of hydrologic alteration. That's sort of interesting, but not really. The more interesting thing is I can go into a watershed. This is the San Diego River watershed, and I can look at the degree of hydrologic alteration in terms of what's important to the biology. So now I've answered the question, how much flow alteration is too much flow alteration? And I can say under contemporary conditions, I've got portions of the watershed where the hydrology is good enough to promote healthy biology, and some of these more urban areas where it's not. But I can overlay the 2050 land use projections for this watershed, and now I can predict in my model how the flow will change under future land use conditions, and I can start to see which catchments are going to start to go from green to orange, or some of these darker colors. And so now I know which areas are priorities for management. So maybe I want to do something differently here. Maybe I want to put in more restrictions or more management measures or more BMPs to try to sort of protect against that degradation. I can also look in this watershed, and I can look at all that other data, all the other stressor data I have. I can ask the question, what sites are in both poor biological condition and poor hydrologic condition? And so those are the sites where flow management becomes sort of the priority in terms of what I need to do as a manager. Other sites, like if the biology is poor but the hydrology is good, that means that probably some other stressor is the problem, right? And so maybe I look at the other stressors and figure out maybe it's something else. These are obviously both good. We want to protect those. And these, the hydrology is poor, but the biology is okay, which either means that A, it's a resilient site, so maybe it's a bedrock-controlled stream or something like that, and so it's more resilient, or maybe it just hasn't responded yet. Maybe it's not far enough onto the response trajectory that it's showing up in the biology yet. So I want to monitor those and watch those a little more closely. So we can use this combination to start to inform management actions. And then the last piece is we can go into sites where there's a scenario where we're looking at like stormwater management. And so in this case, in this Alvarado Creek, we have the contemporary hydrology under a 50% impervious cover. I can go in and I can look at what the natural hydrology would be. And I can say, for some of my key flow metrics, what is the threshold, what's the target that I've set, and how much, if I put in a bunch of BMPs to try to reduce the effective imperviousness down to a level that's closer to natural, how far do I have to go down that trajectory to get to a point where my metric is going to actually hit my target? So for some metrics, I have to really dial down the effective imperviousness, so put in a lot of flow management to get pretty low. But for other things, I don't have to do quite as much, right? And so you can use this to sort of game out different management scenarios and say, how much management do I need to put on the landscape to get the hydrology to a point where it's going to support a healthy biological community, right? So those are, there you go. Right. See, now I want to make sure you're awake, right? I blanked you out of my take-home messages, right? All right, so my take-home messages from all of this are, so it's kind of, I tried to sort of walk you through sort of the data that we produce and how we sort of use that to create these flow ecology relationships, and then how we use those flow ecology relationships to then inform management decisions. So those are sort of the 3 steps, right? All starts with data, as always, right? So the take-home messages is that we know that flow alteration and geomorphic response is really one of the key stresses on our landscape. I would argue that those cute little bugs are a powerful assessment tool, and so we ought to keep investing in measuring them. Regional monitoring, this is my big, anybody who knows me know this, it's impossible for me to give a talk and not talk about regional monitoring, because I can't emphasize how important it is to invest in these large data collection efforts. So just absolutely the things that you can't do if you don't have them are, really hampers you. So it's really critical to have these robust and ongoing regional monitoring programs. Management, you have to work at multiple scales, both the watersheds and the site. You have to have planning and response. And we still have a long way to go in terms of really being able to tease out, this is all associative, right? Everything I told you is statistical. And so in terms of really understanding mechanistically how flow interacts with the physical habitat and the morphology and really teasing out those mechanistic responses, we still have a lot more work to do. So as we look to the future, we're working with folks here at the Center for Watershed Sciences and other places to expand this work, look at other biological endpoints, fish, algae, amphibians potentially. So we want to build this out and look at other biological endpoints. We want this third bullet is something that I'm particularly interested in, is this issue of shifting baselines, right? And as our climate changes, that means our reference expectations also change. And so how does that affect the decisions we make about appropriate hydrology, because our baseline expectations are shifting over time? And then how do we continue to incorporate these tools into policy decisions that the state is making on an ongoing basis? So with that, I'll take any questions. Jay.
I really like your use of bugs. Because it allows you, because bugs are so much cheaper than fish
Right.
to monitor and to study over the year, and so many different places. It allows you to develop a much larger regional view and much more frequent observations if you decide you need to. Within a budget.
Right.
You know, because we would all like all data on everything at all times, in all places. And that just ain't gonna happen.
Right.
So I think it's a real challenge for the state to see, you know, how much can we get out of it. If we can make the problem is most people don't care about bugs.
Right.
I know that's
Yeah.
sacrilege in some cases, some areas, but most people care about fish. So if you could, have you had success in correlating bugs with fish?
Yeah, so it's a really good question, and so I'll answer it in two ways. So yeah, because for a lot of people, bugs are just fish food, right?
Yeah.
And so so I think that there's two ways that I would answer it. There is, there's a lot of what I would call location-specific work that's been done that's really sort of, I think, documented really well how changes in the bug community relate to changes in the fish community, because there's really tight coupling between the bug community and the fish community. So I think there's some knowledge of that. We haven't really incorporated it into this type of work because, again, it's very associative. I think that a lot of the work that we're contemplating doing moving into the future is twofold. One, looking at the relationships between flow alteration and fish, and how you manage sort of the ecohydrology relationship for fish, and how you manage it for bugs, and how you manage it for algae. And then, again, as you'd appreciate, there's a sort of an optimization issue in there, right? Like how do you...
There's always an
Right, exactly, right? So how does flow alteration for, is it important to bugs compared to fish, and is there sort of a way to optimize those two? But I think even more interestingly than that which is again a sort of a more of a statistical type of approach, because we have more computational ability now, I think we're able to start getting into some more of these food web models. And so there's been some really interesting work starting to look at some of these food web models where you can actually start to incorporate things like trophic structure and path length within the food web, and start to look at those types of relationships and build them into these. So you have like a response. Your response variable might not be bugs or fish, but it might be length of the, trophic length or network density of the food web. So I think there's really some interesting emerging work in that arena that may allow us to kind of go down that pathway potentially, which is really exciting because I think we haven't been able to do a lot of that in the past, mainly because of computational limitations, right? And so, yeah.
So I have to ask,
Yeah.
what's your perspective on the LA River revitalization project? I know it's only 11 miles, but I'm sort of curious about whether there is value in the ability to captivate the public imagination for the policymakers or managers for projects upstream of geomorphology or hydrology.
Yeah. Well, I think you answered your own question, right, about the LA, because, the thing with the LA River, right, which most of you are familiar with, right, it's where they drive cars down. I think the value of the LA River, so first I would say there is ecological value in the LA River at there's areas like the Glendale Narrows and the Sepulveda Basin, which actually we know have really important rookery habitats, and they have the potential to provide some important ecological benefits if you change the way they're managed slightly. So there is ecological value there. Of course, it's nothing like what you would expect in a natural river. But in my opinion, the real reason to pursue something like the LA River revitalization is less about the ecology and more about the sociology, right? It's more about creating a better appreciation and an understanding for river ecology and what it means and how the watershed works as a whole. And so to have a concrete channel behind a gate, you just lose that opportunity. So in my opinion, the benefit is more a social benefit in terms of creating more awareness and more stewardship of an understanding of rivers and watersheds than it is, you won't spend $1 billion, based on an ecological argument. I think it's more of sort of a social argument. That's my opinion on why you would do that. So.
Can you talk a little bit about the pros and the cons of algae as an indicator of hydrological
Yeah.
We talked a little bit earlier, the taxonomy is very difficult, but in terms of their sensitivity of response compared to something like quackitza.
Yeah. So it's a really good question. So and I mentioned earlier that we like to use them in concert. So I would say that so algae are really interesting in the sense, and we sort of, for example, there are some diatoms that have certain sort of motility characteristics. And so there's some really interesting adaptations in the algal community to changes in flow. So you can actually see in the algal community that certain taxa will either be more prevalent or less prevalent under certain types of flow or sedimentation conditions. So they can improve your diagnostic ability. But I think the real value of algae is that when we use algae and bugs in concert, as a general rule, I mean, as a sort of a gross generalization, the algae tend to be more responsive to a lot of the water quality parameters. And so we see much tighter coupling with the algae to things like nutrients and some of the temperature variables. And so they tend to be a little more tightly coupled with water quality, whereas the bugs tend to be more tightly coupled with the flow and the habitat characteristics and things like specific conductance. And so using them together is really powerful because, you know, in these urban landscapes or agricultural landscapes or any of these altered landscapes, no stressor occurs in isolation, right? And so you never just have flow alteration in the absence of some other stressor, right? Or it's very rare, right? And so using the two biological indicators together allows you to better sort of separate the proportional contribution of, say, something like flow alteration versus, say, nutrient enrichment. And so I think that's the value of using them together, is because they tell you sort of different things about the system, and using them together can help you sort of tease apart the relative contribution of the different stressors. The other thing that I would say, the other two things I like about algae is they're a little less dependent on substrate. And so when you have so especially sort of hardened channels where you've really kind of changed the substrate, the algae tend to be a little less, little more pliable or plastic as it relates to the substrate composition. So they're a little more resilient to that factor. And they're a little bit, the response is a little sooner. So we often see responses in the algal community a little sooner than in the bug community. And so like earlier today, we were talking about, you and I were talking about some of the molecular methods we're using. And so if you could start to measure the algae more frequently, you might be able to detect responses a little bit sooner, particularly following like a large catastrophic event or in a recovery, like if you're restoring a site and you're looking for recovery. So I think there's lots of advantages of using the two together, but the biggest one is probably the fact that they respond to stressors in different ways. And so using them together gives you a little bit more diagnostic capability. So, Jay? Oh.
I actually have two technical questions.
Yeah.
So you compare the vertical versus horizontal
Yeah.
change. Did you go back to that one?
Yeah.
Because I mean, in many parts of the world, I see more vertical.
Mm-hmm.
Is vertical incision and then a gradation. So
Yeah.
More, but then I was also kind of fascinated about the horizontal change. That one.
Yeah.
So, I don't know, a little bit, I was trying actually to put together how that can happen in, for instance, in the vertical. There is not that much vertical.
Mm-hmm.
We have good conditions or low susceptibility in some of those.
Yeah, I think the basic story here is the story of geology and substrate, right? Because we have very little clay in our systems, right? And they're really just sand, right? So our channels are really just sand. And I think that's really the story here, right? Because I think if we tend to see more of the vertical incision in more consolidated substrates. Yeah, and so I think that's the real story
And then
here.
the lateral is because you change the hydrograph for a mild peak flow
Yeah.
to flashiness. So
Right.
basically they go back full.
Yep.
And that's why they are altering all the vertical
Yeah, and
cross-section.
exactly. And we have these really sandy, sort of sugary types of channels. And so, yeah, they just basically, if they're not constrained by a levee or some hard structure, they're just going to go wide, right? And so that's usually the first vector of response is sort of in the lateral. If they're unconstrained, then they're going to tend to respond to that. Again, I think that's just a function really of the fact that they're really sandy alluvial types of systems.
Sure, and then the chloride and the total phosphorus as part of the 6
Mhm.
main stressors. So chloride, yeah, you put, yeah, that one, the stressor. So I was kind of worried that chloride and phosphorus
Yeah.
as part of the priority stressor. So chloride is mostly because of treatment plants.
I think the chloride is partly that, but it's also I'm not sure. I mean, chloride. What I would say about the chloride is what you don't get in this plot is the spatial aspect. I do have versions of this that I show on maps, and so what we tend to see in Southern California is we have these chloride hotspots in certain regions because of some of the land use practices. So it's in the region, it's sort of higher up, but if you looked at it on a map, what you would see, it's spatially sort of spotty. And phosphorus, I think a lot of that is also associated with some natural weathering patterns also, right, in the substrate. Or Jay or Sarah? Oh.
eDNA, do you see a long-term value or potential growth in use of that, or how promising?
Yeah, so Rob and Sarah and I were talking about this over lunch today. So I'll give the quick answer is I would say yes. We've been doing a lot of work, I would say, over the 10 years as well as all over the world, right? It's kind of a really blowing up this field. And I think so there's different types of, there's eDNA and there's sort of barcoding. I sort of differentiate the two. But just to give you sort of a quick answer, the concept of using molecular methods like to augment the traditional taxonomy, so to identify the species based on extracting the DNA and comparing the DNA to a reference library, I think that is likely to continue to grow. There's a lot of technical issues that still need to be worked out. There's a lot of issues of how do you adapt our existing indices that were built on traditional taxonomy to molecular taxonomy. But I think the reality is we have limitations in the number of taxonomists that are out there. If you go to a room full of taxonomists, they're all sort of near retirement, right? Not a lot of people get trained in taxonomy, especially in algal taxonomy. And these methods are being used more and more in a regulatory context. So I think there's a lot of things pushing us towards, over time, moving towards molecular methods as a tool to help address some of the taxonomy issues. I think they also have some advantages in terms of taxonomic resolution. You can sample more frequently because the turnaround time to get the data is less. It's cheaper, right? So I think there's a lot of things that are pushing the science forward in that area.
How about the water, getting the eDNA from just a water sample as opposed to species?
Yeah, so I think the way we typically do it, because we have an ongoing program in that, particularly the algae, we usually extract it, like for the algae, we filter the algae, or from the bugs, we do the bug in the blender where we blend up the bugs and extract the DNA off the bulk sample. So that, I think that it works pretty well. The DNA from the ambient water, where we take a water sample and try to extract the DNA from the water, so there's a lot of work going on there as well. I would say right now, that's better. The better application of that is for what I would call targeted surveillance. So if you're looking for a specific organism, like a sensitive species or an invasive species, and so you know what you're looking for, and then you can sort of have a primer for that and just look for that, I think it works pretty well. For whole community analysis, I think there's a lot more work to be done, and it's primarily because we don't fully understand the fate of DNA in the ambient environment. So we have to understand all the kinetics behind photodegradation and absorption and bacterial scavenging, things like that, in order to interpret what that signal means, right? So that's sort of where I think we are on that. So, Sarah.
Sorry, I'll hold my question.
Okay.
Okay, thanks so much, Eric.
All right, thanks.